Good evening, and welcome back to SleepWise. Tonight, we turn inward, not toward thoughts or memories, but toward something quieter, older, and more deeply hidden. Inside nearly every cell of your body, folded so delicately and packed so tightly that it disappears from sight, there rests a long molecular thread carrying instructions shaped across an almost unimaginable span of time. It does not speak in words, and yet it preserves patterns. It does not think, and yet it remembers. Long before anyone gave it a name, this hidden code was already at work, guiding growth, passing traits onward, allowing life to remain recognizably itself, while never repeating any living thing exactly. There is something especially fitting at the end of a long day in resting beside a story like this. Genetics is quiet science. It belongs to slow divisions, soft continuities, and invisible repetition. It belongs to the way a face can carry an echo of another face, the way a seed can hold the outline of a future tree, the way a body, without effort or awareness, follows instructions written long before it was born. And so tonight, there is nothing you need to master. You do not need to hold every term in place. You can simply rest here and let the story unfold at its own pace. And if, somewhere along the way, this kind of drifting inquiry feels like the sort of quiet company worth returning to, then perhaps SleepWise is something to follow, and maybe even something gentle enough to share with someone else whose mind is slow to settle at night. For now, we begin with the simplest shape of the mystery. How does life remember? How does a living thing pass forward not only substance, but pattern? How does a child resemble a parent without becoming a copy? How does the world keep producing roses and foxes and whales and human beings, each one linked to what came before, and yet each one also new? Somewhere beneath skin and bone and leaf and feather, life has found a way to preserve information while still making room for variation. That quiet balance between continuity and change is where genetics begins. And perhaps that is why the subject feels so profound when we linger with it. It is not only about molecules, it is also about inheritance, ancestry, resemblance, possibility, and deep time. It is about the strange fact that every living thing is both an arrival and a continuation, something new entering the world, and something ancient still moving through it. So, as the room grows dimmer and the day loosens its grip, we let the mind drift downward toward that invisible continuity, toward the code life has been carrying, revising, and preserving for billions of years, toward the hidden grammar beneath the living world. And we begin, as humans always began, with simple noticing, with the old sense that life was passing something secret from one generation to the next. For most of human history, that secret remained just beyond reach. People lived beside inheritance long before they understood it. They saw it in households and orchards, in stables, gardens, and fields, in the ordinary repetition of faces and forms. A son might carry the brow of his father, a daughter the eyes of her grandmother. One child might seem to echo an older relative no one had mentioned in years. Something was passing forward, that much was plain, but what it was and how it moved remained hidden. Farmers knew this mystery in a practical way. Across many centuries, they saved seeds from the strongest wheat, the sweetest fruit, the hardiest beans. Herders chose animals with calmer temperaments, thicker wool, stronger frames, or better milk. Dogs were bred for guarding, hunting, speed, or companionship. Long before genetics existed as a science, human beings had already learned that traits could be guided. If certain plants were chosen, the next season might lean in one direction. If certain animals were paired, the next litter might carry more of one quality and less of another. And yet inheritance never behaved with perfect obedience. A trait that seemed strong in one generation might fade in the next. Something absent might suddenly return, as if it had only gone quiet for a while. Resemblance moved through families, but never in a straight line. It skipped, lingered, resurfaced. The old world knew the fact of heredity long before it understood the mechanism. Even philosophers and early naturalists found themselves circling the same puzzle. Why do living things reliably produce their own kind? Why does an oak give rise to another oak and not an elm? Why does every new oak arrive recognizably similar, yet not identical, to the last? The living world seemed full of continuity without sameness, variation without chaos. Nature preserved patterns, but she never copied them exactly. There is something soothing in that old uncertainty. For most of human history, life was observed before it was explained. People watched lambs born in spring, noticed which flowers resembled earlier blooms, which children carried old family gestures in the shape of a smile or the angle of a chin, and all around them the same quiet question persisted. How does life carry information across time? That question would sharpen in the 19th century as science grew more systematic and more confident that even deep mysteries might yield to patient observation. Soon, naturalists would begin asking not only how traits are passed onward, but why the living world itself changes, why new forms appear, why others vanish, and how life, over immense stretches of time, becomes something other than it was. The story of genetics had not yet begun in full, but the ground beneath it was slowly being prepared. Tonight, that old question begins to widen. For centuries, people had wondered how traits passed from parent to child, how resemblance moved through families, and why life repeated familiar forms. But in the 19th century, another question pressed forward beside it. It was not only about how life remains itself. It was about how life changes. Charles Darwin was not the first to notice variation in the living world, but he saw it with unusual patience and with a mind willing to follow small differences toward large conclusions. As a young man aboard the HMS Beagle, he traveled far from England and encountered a living world that did not feel fixed or finished. He saw mockingbirds that differed from island to island, tortoises whose forms seemed shaped by place, and finches whose beaks appeared fitted to different ways of living. What mattered was the pattern beneath them. If species had been separately created in their final forms, why should nearby islands hold animals so similar, and yet not quite the same? Why should living things appear so well suited to their environments, and yet still carry hints of older pathways and older kinships? Darwin began to suspect that life was not arranged as a static gallery, but as a branching history. The idea came slowly. In every generation, living things vary. No two individuals are exactly alike. Some are a little hardier, a little swifter, a little better suited to the pressures around them. And because more organisms are born than can survive, nature is never entirely gentle. There is competition for food, shelter, safety, and mates. In that quiet pressure, slight differences can matter. Those whose traits suit the moment more closely are more likely to survive and leave offspring. And if those traits can somehow be passed on, then over long stretches of time, the population itself may change. This was Darwin's great insight. Life can be shaped gradually through countless small siftings, generation after generation. Natural selection was not a force with intention. It did not plan. It did not aim. It simply favored what fit, and let the rest fade more often into silence. There was something both majestic and unsettling in this view. Species were no longer fixed. Humanity itself was no longer separate from the rest of life, but part of the same long unfolding story. The living world became historical. Every feather, paw, leaf, and hand was no longer just a form. It was a form with ancestry. And yet, Darwin's theory rested on a mystery he could not solve. If useful variations are preserved, what exactly is being preserved? What passes from one generation to the next? By what hidden means does life carry forward the small differences on which evolution depends? Darwin could describe the shaping of life with extraordinary power, but the mechanism of inheritance remained obscure, waiting quietly beneath his theory like a locked room beneath a house. That question leads to one of the deepest shifts in modern thought. Life is not only chemistry, it is chemistry arranged so that information can be stored, preserved, copied, and used. A stone may endure for centuries, and a river may keep its course for ages, but neither carries forward instructions for making another stone or another river. Living things do. That is part of what makes them so strange. A seed does not merely contain matter. It contains a plan, though not the rigid sort drawn by an engineer. It carries instructions flexible enough to unfold in soil, weather, and time. An embryo does not simply grow because material has been assembled in one place. It grows because cells divide while following patterns, turning some processes on and others off, building difference from repetition. Life persists not by holding one shape forever, but by passing forward organized information that can be read anew. This idea is easy to say now, but it was once revolutionary. For long stretches of history, organisms were described by outward form, habitat, or visible function. But genetics invited a deeper view. Beneath wings and bark and skin lay instructions. Beneath appearance lay code. And code is an unusual thing in nature. It is abstract and physical at once. It must exist in matter. But what matters most is not only the substance itself. It is also the order. Rearranged slightly, the same basic material can mean something different. Here, the living world begins to resemble language or writing. A few simple elements, placed in different sequences, can produce extraordinary complexity. Small changes can alter the outcome. Patterns can be copied, edited, interrupted, repeated. There is something meditative in realizing this. Inside the body, countless events unfold without announcement, yet they are not random. Cells are not inventing the whole of life from moment to moment. They are reading inherited instructions, responding to signals, copying certain patterns, silencing others. The living body is busy, but not chaotic. Beneath its activity, there is an order so old and continuous that each generation receives it already in motion. And yet, information in life is never static. It is preserved, but not untouched. It can be copied faithfully, but not perfectly. It can remain silent in one context and active in another. It can endure for ages and then shift. This is why genetics matters not only to heredity, but to evolution, development, disease, ancestry, and identity. To speak of genes is to speak of memory made physical. So before we even arrive at the specific experiments and names, something essential has already come into view. The living world is not merely built. It is instructed, not rigidly, not flawlessly, but persistently. And somewhere in the quiet investigations of the 19th century, someone would begin to glimpse those instructions not as metaphor, but as pattern, hidden, orderly, and measurable at last. Once Mendel's ideas were finally understood, heredity began to lose its mist and take on contour. The old image of blending, in which parental qualities simply mixed together like pigments in water, could no longer hold. If inheritance truly worked that way, then distinct differences should fade steadily with time. But Mendel had shown something else. Traits could disappear from sight without being erased. They could pass quietly through one generation and return in the next, unchanged in their essential character. This was one of the most calming discoveries in biology. Life did not preserve pattern by vague resemblance alone. It preserved it through units. What Mendel called factors, later generations would call genes. In simple cases, an organism carries two versions of a hereditary instruction, one inherited from each parent. These versions do not always announce themselves equally. One may dominate in visible expression, while the other remains recessive, hidden from view, but still present, still capable of being passed onward. That is why a trait can seem to vanish and then return. It was never gone. It was merely quiet. There is something almost musical in this logic. A note may not be heard in one phrase, yet still belong to the composition. Heredity, in this sense, is less like paint mixing and more like patterns being carried and revealed under certain conditions. Of course, real life is often more complicated than the pea plants of a monastery garden. Many traits do not follow neat, dominant, and recessive patterns. Some are shaped by many genes at once. Some are influenced by environment, development, or chance. And yet, Mendel's simplicity remains precious because it revealed the first clear architecture beneath inheritance. He showed that heredity was not formless. It had structure, order, and recurrence. This also changed the meaning of variation. Differences between offspring were no longer simply mysterious outcomes of blending. They arose because hereditary units could be combined in different ways, hidden for a time, then made visible again. The future was not random. It was patterned, though not always easily read. And so biology acquired a new kind of clarity. A child might resemble one parent more than another in a visible trait, not because the other parent contributed nothing, but because some hereditary instructions remain unexpressed in one generation while continuing their quiet journey forward. The visible world became only part of the story. That distinction matters more than it first seems to. Genetics is full of hidden persistence. What appears absent may still be present. What seems lost may still be traveling silently into the future. Inheritance, once thought to be a blur, had become something subtler and more exact. A quiet logic beneath family resemblance, waiting to be traced further inward, from visible traits to the hidden structures inside the cell. By the end of the 19th century, heredity had become more structured in thought, but the body itself was beginning to reveal its hidden architecture. The microscope, steadily improved, was drawing science inward. What once seemed like undifferentiated living substance began to separate into parts. Cells came into view as the basic units of life. Within them, a darker central region appeared again and again. This was the nucleus, a small chamber inside the cell, and it quickly became one of the most intriguing places in biology. If hereditary instructions existed somewhere in the body, many began to suspect they might be found there. Soon, during cell division, other structures became visible as well. Fine material in the nucleus condensed into thread-like bodies that could be stained and seen more clearly under the microscope. These were chromosomes. They appeared, divided, and were passed into daughter cells with a regularity too striking to ignore. Even before their full role was understood, they seemed important. They behaved like carriers of order. There is something deeply satisfying in this stage of the story. What had once been guessed at in families and gardens was now beginning to acquire a home. Inheritance was not just an abstract principle drifting through life. It seemed to reside in structures inside particular cells. Gradually, the connection deepened. Organisms inherit chromosomes from both parents. During reproduction and development, these structures help preserve continuity while allowing recombination and variation. The old mystery of heredity was narrowing. Life was not carrying its patterns through the body in a vague, general way. It was using physical structures housed in cells arranged with remarkable consistency. And yet, a chromosome was not the final answer. To know that these thread-like bodies mattered was not the same as knowing what exactly within them held the hereditary instructions. Chromosomes were clearly central, but they still concealed a more intimate level of order. What material inside these structures actually carried the code of life? The search for that answer would take time because the candidates did not seem equal. Proteins, with their many shapes and varied functions, looked rich and promising. Another substance, present in chromosomes and eventually named deoxyribonucleic acid, seemed at first almost too simple to matter. It lacked the glamour of proteins. It looked repetitive, perhaps structural rather than instructive. Science was now close enough to touch the real material of heredity and still far enough away to misunderstand it. That is often how knowledge advances. By now, the hidden thread of inheritance had been traced into the cell, into the nucleus, into the chromosome. The house of heredity was becoming visible room by room. But the deepest chamber had not yet been opened. Somewhere inside those quiet structures, the code itself was waiting. When scientists finally turned more closely toward DNA, it did not at first look like the molecule that would explain life. It seemed too modest for that burden. Proteins appeared far more elaborate, with their varied forms and many roles in the body. DNA, by contrast, seemed repetitive, almost monotonous. Few would have guessed that this quiet substance tucked inside chromosomes would become one of the most important molecules ever studied. And yet, slowly, the evidence shifted. Experiments in the first half of the 20th century began to suggest that hereditary information could be transferred through a substance that was not protein. Certain traits in bacteria could be changed by exposure to material from other cells, as though instructions themselves were moving from one organism to another. The implication was extraordinary. Inheritance was not merely associated with DNA. It might actually reside there. Once that possibility took hold, the molecule itself began to demand a new kind of attention. What was DNA made of? How could something so seemingly simple preserve such immense complexity? The answer, when it emerged, would depend on structure. DNA is built from repeating units called nucleotides. Each contains a sugar, a phosphate group, and one of 4 chemical bases. Those bases are adenine, thymine, cytosine, and guanine. 4 small components arranged in sequence along a molecular backbone. There is something almost impossible in the elegance of that arrangement. Only 4 letters, and yet from them the living world composes orchids, foxes, lungs, coral reefs, hands, and minds. What matters, then, is not just the material, but the order. DNA stores information because the sequence can vary. A different order means a different instruction. Here, life begins to resemble writing in the deepest possible sense. A few simple symbols, placed in differing patterns, can preserve and transmit astonishing complexity. And still, one crucial question remained. How could such a molecule be copied? How could hereditary information pass so faithfully from cell to cell and generation to generation unless the structure itself made copying possible? Scientists now knew that DNA mattered. They knew it occupied the right place and possessed the right kind of informational capacity. But they had not yet seen its full form. The answer would arrive through a convergence of chemistry, physics, and patient imaging, and when it did, the molecule would appear not as a tangled chain or a shapeless thread, but as one of the most graceful structures in science. DNA was not merely a list, it was an architecture. So the story pauses here, on the edge of revelation. The material of heredity has come into view. 4 chemical bases wait in silence. The sequence matters. The code is real. And somewhere, hidden in the geometry of the molecule itself, lies the secret of how life remembers by copying. Once the double helix had come into view, the molecule seemed at once stranger and simpler than anyone had expected. Life, in all its abundance, did not rest on a library of endless symbols. It rested on 4 chemical bases, repeated in different orders along the length of DNA: adenine, thymine, cytosine, guanine. 4 small components, modest in number and yet capable, through sequence alone, of carrying instructions for bodies, lineages, adaptations, and the slow remembering of the living world. There is something deeply calming in that restraint. So much of existence seems to unfold from extravagance. Forests without number, animals without count. The unrepeatable details of faces, hands, feathers, leaves. And yet, the molecular alphabet beneath all of this is remarkably spare. 4 letters only. Not because life is simple, but because simplicity, repeated with order, can become inexhaustibly rich. What matters, then, is arrangement. A different sequence means a different instruction, just as the same handful of musical notes can produce one melody or another, depending on their order. DNA does not need thousands of separate ingredients. It needs a stable alphabet and a way of placing its symbols into meaningful succession. The code of life is written not through variety of letters, but through variation of sequence, and the sequence does not float alone. Each base is paired with its quiet partner. Adenine joins to thymine. Cytosine joins to guanine. That pairing is one of the most elegant rules in biology. It means that one strand of DNA implies the other. Half the molecule contains, in a hidden way, the logic of the whole. If one side is known, the matching side can be reconstructed. The code is not only stored, it is mirrored. This is why the double helix is such a beautiful scientific discovery. Its form is not merely decorative. The structure explains how information can be preserved with such steadiness. DNA is a sequence, but it is also a relationship. One strand calls quietly to the other, base by base, letter by letter, across the spiral. There is, too, something humbling in the fact that such grandeur depends on rules so narrow and exact. No base drifts toward just any companion. The molecule holds because its pairings are precise. Fidelity in life begins here, in small acts of chemical fit repeated over unimaginable spans of time. And so the old mystery of inheritance grows clearer still. Traits endure because instructions endure. Instructions endure because sequences endure. And sequences endure because The molecule that carries them is built with an internal logic gentle enough to persist and exact enough to be copied. Once that became clear, one further wonder moved to the center of the story. If each strand contains the key to rebuilding its partner, then how does life actually perform that copying? Cell after cell, generation after generation, without losing itself along the way? Replication is one of the quiet miracles of the cell. It happens without applause, without sensation, and without the mind ever knowing it has occurred. Yet every time a cell prepares to divide, the long DNA molecule must be copied with extraordinary care, so that each new cell receives its own inheritance. Growth depends on it. Repair depends on it. The continuity of the body and the continuity of life beyond the body depend on it. The process begins with separation. The two strands of the double helix part from one another, not violently, but with a delicacy that suits the scale of the event. The old paired bases pull apart, and the molecule opens. What had seemed like one structure becomes two templates, each carrying the information needed to guide the making of its missing partner. Then the quiet rule of pairing returns. Adenine draws thymine. Cytosine draws guanine. Along each exposed strand, new complementary bases are assembled in sequence, and the old information is used to generate a fresh copy. In the end, two DNA molecules stand where one stood before. Each contains one older strand and one newly built strand, a lovely compromise between preservation and renewal. Life does not keep the past by freezing it. Life keeps the past by rebuilding it. There is something especially soothing in that thought. Your body is never perfectly still. Cells are lost, replaced, repaired. Materials move through you. Yet form persists because pattern persists. The body remains recognizably itself, not by holding all matter unchanged, but by maintaining the instructions through continual acts of careful copying. Of course, replication is not absolutely perfect, and that matters too. If it were careless, heredity would dissolve into noise. If it were flawless beyond all exception, variation would have little room to enter. Instead, life inhabits a remarkable balance. Copying is accurate enough to preserve species, tissues, and continuity, but not so inflexible that change becomes impossible. Most of the time, the code passes onward almost unchanged. Occasionally, a small difference slips through. Much later in the story, evolution will depend on that slight imperfection. But for now, what matters is fidelity. Replication gave genetics a rhythm. Inheritance was no longer a vague tendency or a mystery carried through blood alone. It was a repeatable cellular process, grounded in structure, governed by pairing, and performed incessantly in the dark interior of living tissue. And once the code could be stored and copied, another question pressed forward. DNA may preserve instructions across time, but a body is not built by storage alone. How does the cell decide which parts of the code to use? How does a long archive become living action? That question leads to an important softening of the genetic story. It is tempting, When first learning about DNA, to imagine the genome as a tidy row of commands, each gene linked neatly to a trait, each instruction waiting to unfold in a direct line from code to outcome. But the deeper one moves into genetics, the more spacious and subtle the picture becomes. A gene is real, and genes matter immensely. Yet the genome is not merely a list of simple orders. It is a vast arranged landscape of instructions, signals, pauses, boundaries, and quiet regions whose roles took science years to appreciate. Some stretches of DNA contain instructions for building proteins, and for a long time, those regions drew most of the attention. It was natural to assume that the most important parts of the genome would be the ones directly translated into the machinery of the cell. But over time, biology learned that much of the genome is not best understood as a row of protein recipes. Some sequences regulate when a gene is used, where it is used, and how much of it is read. Others help shape structure, timing, or stability. Some are repeated like refrains. Some sit silently for long periods, their significance only gradually coming into view. This does not make genetics less elegant. It makes it more mature. The genome is not a crude switchboard. It is closer to a score, where notes matter, but so do rests, tempo, emphasis, and the moment at which one instrument enters while another remains silent. Meaning lies not only in the presence of an instruction, but in the conditions under which it is read. There is a comfort in leaving behind the simpler fantasy that one gene always equals one visible feature. Living things are more layered than that. The body is not built from isolated commands firing one by one. It emerges from networks of instructions interacting over time within cells that respond to signals, histories, and contexts. So when we speak of genes, it is best to do so with both clarity and modesty. A gene is a real unit of hereditary information, but it lives inside a broader genomic world. It is part of a system, not an island. That is why genetic knowledge became more powerful as it became less simplistic. The more closely science looked at the genome, the less tempting it became to speak in absolutes. Biology did not grow blurrier. It grew more truthful. The living world was not powered by a single layer of instruction, but by layers nested within layers, order braided with timing. And this prepares the way for one of the most beautiful transitions in all of biology. DNA may hold the archive, but archives do not act by themselves. Somewhere between stored sequence and living tissue, the cell must create a working copy, carry it outward, and turn molecular information into form, motion, and change. To understand how a gene becomes part of a living body, it helps to imagine a quiet library that does not lend out its rarest book. DNA remains stored in the nucleus, protected as an archive of hereditary information. But when the cell needs to use a particular instruction, it does not usually drag the original volume into the workshop. Instead, it makes a temporary copy. That copy is RNA. This first stage is called transcription. A selected stretch of DNA is read, and its sequence is copied into a related molecule, one light enough to travel, one suited not to long-term storage, but to immediate use. If DNA is the durable memory of the cell, RNA is the message carried from memory into action. Already, the system shows its quiet wisdom. The archive is preserved while work proceeds from a temporary transcript. Then comes translation, a name that feels exactly right. The RNA sequence is read in small groups, and those groups correspond to amino acids, the building blocks from which proteins are made. One by one, amino acids are linked into a chain. That chain then folds into a particular form, and its form helps determine its role. Some proteins build structure, some carry signals, some move materials from one place to another. Many act as enzymes, guiding chemical reactions with astonishing speed and precision. Much of what the cell does, it does through proteins. There is something almost serene in the layered nature of this process. DNA to RNA to protein. Archive to message to function. Nothing is rushed and nothing appears all at once. The cell moves through stages, handing information from one form to the next until hereditary code becomes living activity. And yet, even here, genetics refuses to be simplistic. Not every gene is used in every cell. Not every RNA message is made at the same moment. Not every protein appears in equal quantity. The life of the cell depends not only on what is possible, but on what is chosen, timed, and regulated. It also helps explain why heredity can feel both ancient and immediate. The same chain of information that links you to distant ancestors is also being transcribed and translated within you now, in cells, quietly making the molecules required for repair, maintenance, growth, signaling, and thought. There is also a quiet economy in the design. The cell does not expose the full archive every time it needs a task performed. It copies only what is needed, in the amount required, and often only for a while. In that restraint lies another kind of elegance. Genetic information is not merely stored and unleashed. It is managed with patience. Once you begin to see this, a further question arises almost by itself. If nearly every cell contains the same DNA archive, why does one cell become nerve, another skin, another muscle, another blood? How can a shared script give rise to such different forms of life within one body? That question opens into one of the gentlest and most profound ideas in genetics. Nearly every cell in your body carries essentially the same genome, and yet the body is not a field of identical cells repeating one pattern without variation. A neuron does not behave like a liver cell. A muscle fiber is not a blood cell. The tissues of the eye are not the tissues of the heart. The inherited script is largely shared, but the reading of it is selective. Here, the genome begins to resemble music more than machinery. The score may be the same, but not every instrument plays every line. Some genes are active in one cell type and quiet in another. Some are switched on briefly during development, then dimmed. Others remain silent for years, unless a particular condition calls them forth. Difference in the body emerges not because each cell carries a wholly different book, but because each cell reads from the common book in its own way. Part of this selectivity depends on regulation so delicate that biology took a long time to appreciate its depth. Chemical marks and structural changes can influence whether certain regions of DNA are accessible or tucked away, active or muted. This is part of what is meant by epigenetics. The word sounds heavy, but the idea is subtle. It usually does not rewrite the underlying sequence. It influences how the sequence is used. The text remains, but its reading is shaped. There is something deeply reassuring in that. Genetics is powerful, but it is not a simple tyranny of code. DNA matters enormously, yet life is also governed by timing, context, development, environment, and layers of regulation that shape which instructions are brought forward and which are allowed to rest. The body is not a blunt execution of one fixed plan. It is a living interpretation, responsive and layered. This is why the same genome can help form both continuity and difference. It links every cell in the body to a common origin, yet allows those cells to become meaningfully distinct. Identity in biology is not only a matter of what is present, it is also a matter of what is expressed, when, where, and to what degree. And this is where genetics becomes more humane as well as more precise. It leaves room for individuality without denying inheritance, and for biology without collapsing life into rigid fate. The body remembers, but it also responds. It carries instructions, but it does not live by instructions alone. And once that becomes clear, the old story of heredity begins to widen again. If DNA can be copied with immense fidelity, yet not perfect fidelity, if instructions can be shared yet expressed differently, if the same code can sustain both constancy and variation, then small changes in sequence may matter differently depending on where they arise and how they are read. In that small opening lies variation, and in variation the great evolutionary story quietly returns. Darwin had changed the mood of biology. The living world was no longer a still collection of forms, each species fixed in place like an object in a cabinet. It had become a moving history, a long unfolding, full of branching, adaptation, loss, and return. Creatures were linked not only by appearance but by descent. The finch, the fox, the orchid, the hand, each now belonged to time. And yet, beneath that vast new vision, there remained a quiet uncertainty, one Darwin himself could never fully dissolve. He could describe, with remarkable patience and force, how favorable traits might become more common. He could show how slight differences, preserved across many generations, might gradually alter an entire population. But he could not say with confidence what exactly was being passed onward. By what hidden means did life carry its successful variations into the future? That question mattered more than it first seemed. Natural selection could shape life only if life possessed some way of remembering. A useful trait must do more than appear once. It must survive reproduction. It must be carried intact enough from parent to offspring and then onward again. Without that continuity, change would blur as quickly as it arose. Evolution required not only variation, but inheritance with structure. In Darwin's century, many people still imagined heredity as a kind of blending. It felt intuitive. The qualities of mother and father seemed to mix in the child the way colors might mix in water. But this picture held a problem. If inheritance were only blending, differences should steadily soften with time. Distinct traits would fade into averages, and the sharp edges of variation would slowly disappear. Yet nature did not look softened in that way. Traits vanished and returned. Family likeness could skip a generation, then reappear with surprising force, as though something had remained hidden rather than lost. So there, beneath the grandeur of Darwin's theory, lay an unopened chamber. He had shown that life changes. He had shown that the pressures of the world quietly sift those changes. But the thing being sifted, the underlying units of heredity, the means by which the past remained present, still had no clear form. There is something almost comforting in that incompleteness. Science rarely arrives all at once. One mind sees the movement before another sees the mechanism. Darwin gave the world its great biological panorama, but heredity remained dimly lit, still waiting for someone patient enough to notice that life was carrying not just substance, but pattern. And so the question deepened. How does the future receive the past? How does life preserve difference without dissolving it? Somewhere, beneath resemblance, beneath breeding, beneath the broad sweep of evolution, there had to be a hidden grammar. Something quiet, stable, and real, passing from one generation to the next. Long after Darwin, when genes had begun to emerge from the fog of heredity, another voice helped people feel the strangeness of the subject in a new way. Richard Dawkins did not uncover the first laws of inheritance, nor discover the structure of DNA, but he offered a perspective that settled into the modern imagination with unusual force. He asked, what happens if we shift our attention slightly away from the organism alone and toward the hereditary instructions moving through it? At first, the idea can feel a little cold, almost too abstract for the warmth of living things. We are used to beginning with the animal, the tree, the human life, with bodies that breathe and age. But Dawkins invited the mind to notice another time scale beneath that familiar one. A fox lives one life. An oak stands for years. A human being enters the world, changes, loves, remembers, and departs. Yet the hereditary patterns carried within those lives may continue if they are passed onward. In that sense, genes can be imagined as quiet travelers in time. They do not move with intention, and they do not possess wishes of their own, but they endure by being copied. Bodies are the brief and beautiful arrangements through which that copying becomes possible. One generation shelters the code for a while, then hands it onward, and another generation receives it. Life, seen from this angle, is full of continuity hidden inside transience. What makes this perspective so powerful is not that it replaces the visible world, but that it deepens it. A bird remains a bird. A person remains a person. Nothing meaningful is lost by saying that inherited instructions also persist through them. If anything, the world grows more layered. Every organism becomes both itself and a meeting place for deep history. A present form carrying patterns that have traveled through many lives to arrive here. And yet this way of seeing must be held gently. Genes are not little tyrants ruling the organism from within. They act in cells, in bodies, in environments, and among countless other processes. The gene's eye view is valuable not because it explains everything, but because it clarifies one extraordinary truth. Life remembers by preserving information through reproduction. That truth prepares the mind for the next step. If hereditary patterns really can remain stable while bodies come and go, then inheritance is not a mist or tendency. It has structure. Somewhere in the history of science, someone must have glimpsed those hidden units clearly enough to trace their behavior. Somewhere, the old mystery of family resemblance must have begun to sharpen into rules. And that sharpening, when it came, did not begin in a grand laboratory or aboard a famous ship. It began in stillness, in patient counting, in a garden where traits appeared, disappeared, and returned with such quiet regularity that heredity itself began at last to show its hand. Before heredity could be seen, it had to be felt in a new way. For most of human history, people noticed likeness. They saw parents echoed in children, old family features resurfacing after years of silence, and the recognizable forms of swallows, wolves, and apple trees continuing across generations. But noticing likeness is not yet the same as understanding inheritance. A resemblance can be admired without being explained. What changed in the 19th century was the suspicion that living things were carrying something more precise than resemblance alone. Life was beginning to look as though it possessed memory. Not memory in the human sense, not recollection or thought, but a quieter continuity by which form could survive the passing of individual bodies. A leaf falls, a deer ages, a human life comes and goes, and yet the pattern does not vanish with the individual. Something crosses the threshold from one generation into the next, stable enough to preserve a species, flexible enough to allow variation, hidden enough to escape the eye. There is something almost trance-like in resting with that idea. The living world does not simply continue because matter keeps reappearing in roughly the same shape. It continues because some underlying order is transmitted. A seed carries more than substance. It carries arrangement. An embryo unfolds not through random accumulation, but through inherited instruction. The body is not only built, it is guided. Beneath bloodlines and branches and breeding, there seems to lie a quiet script, not yet read, not yet located, but already leaving its trace in the repetition of life. And once that thought takes hold, the old mystery changes character. Heredity is no longer a soft atmosphere surrounding family resemblance. It becomes something firmer and more demanding. If life truly preserves hidden order, then that order ought to reveal itself somehow. The invisible should make itself known through the visible. If only someone were patient enough to watch generation after generation and notice what returns, what disappears, and what was never truly lost. Before anyone could name genes or see chromosomes or imagine DNA, there first had to be the confidence that heredity possessed structure at all. Biology was learning to believe that beneath the flowing variety of life, there must be hidden architecture that preserved difference without allowing it to dissolve into blur. Darwin had shown that life changes. The idea now gathering strength was that life also remembers, and that this memory might be measured. And when the measurement finally came, it did not arrive as thunder. It came through repetition, patience, and counting in a monastery garden. Genetics was waiting there, folded inside ordinary things, round seeds and wrinkled ones, tall stems and short stems, visible traits quietly revealing invisible order. At last, what Mendel uncovered was not merely that traits pass from parent to offspring. His deeper discovery was that heredity can carry something quietly, even when the eye no longer sees it. A trait may disappear from view without disappearing from the lineage. It may pass through one generation in silence and then return in the next with its identity intact. That possibility altered the whole atmosphere of inheritance. When Mendel crossed pea plants with contrasting traits, the first generation often seemed deceptively simple. One trait appeared plainly, while the other seemed to have vanished. Tallness remained visible. Shortness fell quiet. Smooth seeds appeared, wrinkling withdrew. Had heredity worked only by blending, that might have been the end of the story. The two parental forms would have merged into an average, and distinction itself would slowly have washed away. But Mendel kept going, and in the next generation he saw the hidden trait return. Not blurred, not weakened, returned. That is why his work was so revolutionary. Hereditary influences do not melt into one another like pigments stirred in water. They remain discreet enough to be preserved, even when not expressed. One version of a trait may dominate in outward appearance, while another remains recessive, present but concealed. Hidden does not mean absent. Quiet does not mean gone. Nature, Mendel showed, often carries more than she immediately reveals. The living world is not always transparent on the surface. Some possibilities travel in silence. A family line may carry a feature that skips a generation only to appear later, as though the past had been waiting for the right moment to speak again. Today, we describe this with the language of dominant and recessive inheritance. In simple cases, an organism receives one hereditary factor from each parent. Sometimes, one of those factors determines the visible trait, while the other continues its journey unseen. The visible world, then, is only part of the genetic world. Beneath what appears lies another layer of continuity, folded inward, still traveling. Of course, the deeper genetics goes, the more complexity it finds. Many traits are shaped by many genes, and many outcomes depend on environment, development, and timing as well. But none of that diminishes Mendel's significance. He gave heredity its first dependable grammar. He showed that inheritance has architecture, that variation can be preserved without chaos, and continuity maintained without sameness. For Darwin's changing world, This mattered profoundly. The differences on which natural selection acts need not dissolve from one generation to the next. They can be carried, masked, recombined, and later revealed. The next question, then, was irresistible. If hereditary factors behave as stable units, where in the living body are those units held? Now, the old evolutionary story returns, but in a new and more intimate form. Darwin had seen that life changes across generations, and Mendel had shown that inheritance follows patterns. But DNA reveals where variation actually enters the stream. It enters through change in the sequence itself. A base is replaced, lost, repeated, or rearranged, and the inherited script becomes slightly different from what it was before. Many of these changes do almost nothing at all. Some are harmful. A few in the right setting become useful. But whether dramatic or quiet, they remind us that heredity is never perfectly still. These changes are called mutations, though the word can sound harsher than the reality. In truth, mutation is simply one of the ordinary conditions of living inheritance. DNA is copied with immense fidelity, but not with absolute perfection. And over great stretches of time, even rare changes accumulate. Sometimes they arise in regions of the genome that matter little to the organism, and their effects pass almost unnoticed. Sometimes they alter a protein, or shift the timing of a gene, or change the way a cell responds to its environment. Sometimes they vanish again. Sometimes they travel forward. Variation does not arrive from outside evolution as a separate force. It is the raw material on which evolution works. Natural selection cannot favor a trait unless there is some inherited difference already present in a population. Mutation provides part of that difference. Recombination, too, reshuffles inherited material into new combinations. Together, they ensure that life does not merely repeat itself, but continues to generate new possibilities. And yet, the process is neither hurried nor theatrical. Evolution usually proceeds through small siftings, not grand leaps. A variation that helps in one environment may matter little in another. A trait that is costly in one climate may become useful under different conditions. The living world is not marching toward perfection. It is adjusting again and again to changing circumstances, carrying forward what fits well enough for the moment. This is why genetics makes Darwin's insight feel both more precise and more tender. Selection is not acting on vague differences floating in the air. It acts on inherited variation grounded in molecules, copied through cells, preserved through reproduction, and tested in the quiet pressure of life. The old branching tree of evolution now has texture at its smallest scale. Each leaf and feather and hand is part of a history written in changes so tiny they can hide inside a single letter of the code. And still, genetics resists simplicity. Not every visible trait comes from one mutation or one gene. Much of what makes a living thing what it is emerges from many small influences interacting at once. And so, having returned to variation, the story must widen once more, beyond simple inheritance, toward the deeper complexity of real organisms. By now, genetics has become both clearer and more complicated than it first seemed. Mendel gave us the grace of simple patterns, and those patterns were real. But once science moved from monastery gardens into the full thickness of life, it found that many of the traits people wonder about most do not rest on a single hidden factor, quietly deciding the outcome. Height, temperament, vulnerability to illness, the pace of growth, the way a body responds to stress or nourishment, these often emerge from many genes acting together and from many influences beyond genes as well. There is something deeply reassuring in that complexity. It means heredity matters without reducing a life to a verdict. A genetic variant may tilt the body in one direction, but a tilt is not the same as a destiny. Many inherited differences are small in effect, and many only matter in combination with others. Some influence how likely a trait may be, rather than whether it must appear. So, the deeper genetics goes, the more measured its language becomes. Not command, but tendency. Not certainty, but risk. Not one cause, but many layered causes moving together. This is why real organisms feel richer than simple diagrams. A person is not assembled from one line of code for height, one for intelligence, one for mood, one for disease, each acting in splendid isolation. Biology is more like weather than arithmetic. Currents crossing currents, inherited patterns interacting with one another and with the conditions of life. The genome matters profoundly, but it is not the whole sky. And yet, complexity does not mean confusion. It means relation. A body develops through networks of influence, where many small differences can accumulate, cancel, strengthen, or soften one another. Nutrition matters, stress matters, early development matters, chance matters, and through it all, genes remain part of the story. Not absent, not all-powerful, but woven into a wider fabric. There is a kind of wisdom in this. The first discoveries of genetics taught us that inheritance has structure. Later discoveries teach us that structure is not the same as simplicity. The code of life is real, but it is interpreted through context. The same hereditary tendency may unfold differently in different environments. Biology keeps its order, but not by becoming mechanical. Perhaps that is why the subject becomes more humane the further it advances. The old fear that genes might function as fate begins to loosen. What emerges instead is something subtler, a picture in which inheritance, development, environment, and time all meet inside a single life. You are shaped by code, yes, but never exhausted by it. And from here, the story widens once more. Once scientists understood that heredity could be both structured and complex, a grander ambition became possible: to read not just one gene or one trait, but the inherited script of an entire species. There was a time when even a single gene could feel remote, hidden deep within the cell, and difficult to isolate with certainty. Then, over the course of the late 20th century, genetics widened into one of the boldest scientific ambitions ever attempted. The goal was no longer simply to study heredity in fragments, but to read the human genome itself, to map, letter by letter, the long molecular text carried within our species. This became the Human Genome Project, and there is something quietly epic in the scale of that effort. Across laboratories, countries, instruments, and years of work, scientists set out to identify and assemble billions of chemical letters into a reference sequence. The task demanded patience more than spectacle. It was less like the flash of a single discovery and more like the slow charting of an immense coastline, where each new stretch revealed not an ending, but another contour of the same vast land. For most of history, heredity had to be inferred from appearances. Then it could be counted in traits, traced into chromosomes, and localized to DNA. Now the species was attempting something far more intimate, to read its own underlying script at scale. It is difficult not to feel some hush before that moment. Humanity had lived inside this code for countless generations without seeing it directly. And now, at last, it was beginning to turn the pages. Yet the genome, once read, did not offer the kind of closure some had imagined. It did not reveal a tiny and final list of instructions for personhood. It did not flatten the mystery of life into a clean formula. Instead, it opened further depth. Some regions were easier to understand than others. Some functions became clearer, while many remained uncertain. Later work would refine the map, close long-standing gaps, and broaden it beyond any single reference sequence. The closer genetics moved toward completeness, the more it discovered both knowledge and complexity waiting together. That, too, feels appropriate. A genome is not a slogan. It is an inherited landscape shaped by repetition, regulation, mutation, deep time, and evolutionary compromise. To read it is not to conquer life, but to enter more deeply into its detail. Still, the achievement changed biology forever. With genomic tools, researchers could compare populations, trace ancestry, search for variants linked to disease, and study heredity with a precision earlier centuries could scarcely have imagined. The science of inheritance became, in a new sense, a science of reading, and once the code could be read with such power, another possibility began to gather at the edge of thought. If the inherited script can be sequenced, studied, and interpreted, might it someday be changed deliberately as well? That possibility belongs to the newest and perhaps most delicate chapter in the story of genetics. For a long time, DNA could be observed, copied, mapped, and interpreted, but not deliberately changed with much precision. Then, gene editing began to shift the horizon. Among the most famous of these tools is CRISPR, adapted from a natural bacterial defense system and refined into a way of targeting particular stretches of DNA with remarkable specificity. The image is almost startling in its simplicity, not merely reading the code of life, but locating a chosen passage and attempting to revise it. There is good reason this idea has inspired so much hope. If certain diseases arise from harmful variants in DNA, then the prospect of correcting those variants seems almost luminous. Conditions long managed but not repaired might be addressed closer to their source. Cells might be altered to resist disease. Therapies might become more exact, more personal, and more durable. For families shaped by inherited illness, the possibility is not abstract. It reaches toward relief. And yet, genetics, at its best, teaches caution alongside wonder. To edit DNA is not like correcting a word on a printed page and setting the book back on the shelf. Genes exist within systems. A change may have consequences beyond the immediate target. Development is layered. Regulation is subtle. The body is full of interactions we still do not completely understand. Precision has improved enormously, but humility remains part of the discipline. There are deeper questions, too. It is one thing to alter cells in order to treat disease within a living patient. It is another to imagine making changes that would pass onward into future generations. The first belongs largely to medicine. The second reaches into inheritance itself. Questions of fairness, consent, access, unintended consequence, and the meaning of human intervention arise very quickly here, and they should. The closer science comes to power, the more carefully it must travel. So CRISPR belongs in this story not as a triumphant ending, but as a threshold. It shows how far genetics has come, from noticing resemblance, to counting patterns, to tracing chromosomes, to discovering DNA, to reading the genome, and now, cautiously, to revising selected parts of the script itself. The code life has carried for billions of years is no longer only something we inherit. In limited and carefully chosen ways, it is becoming something we may intervene in. There is awe in that, but awe is not the same as conquest. The proper mood is seriousness. The living code is powerful precisely because it is ancient, intricate, and never fully simple. And so before the night closes, it feels right to step back from this frontier and gather the story whole, not as a list of findings, but as a meditation on continuity, variation, and the quiet memory inside life. And now, as the mind begins to loosen its hold on names and mechanisms, what remains is a gentler impression. Beneath the visible world, there is continuity. Beneath continuity, there is inheritance. And beneath inheritance, there is a code. Quiet, ancient, endlessly copied, and never entirely still. What we have traced tonight is not only the history of a molecule, but the history of how life remembers. Darwin helped reveal that living things are not fixed, but shaped across immense spans of time. Mendel showed that inheritance has order, that traits can be carried even when hidden. Later, science followed that order inward into chromosomes, into DNA, into a four-letter sequence whose structure allows information to be stored, copied, read, regulated, varied, and now, in careful circumstances, even edited. What once seemed like separate mysteries now belong to one long continuity. And yet, perhaps the deepest lesson is one of humility. Genes matter immensely, but they do not tell the whole story of a life. The genome is powerful, but it is not a person. Sequence matters, but so do timing, development, environment, chance, and the long layered conversation between inheritance and experience. The deeper genetics goes, the less crude it becomes. It does not flatten the world. It reveals how richly the world is composed. There is comfort in that. To learn that you carry ancient code is not to learn that you are trapped inside a script written long ago. It is to learn that you belong to a continuity older than memory and wider than any single life. The same logic that shaped forests and finches, moss and whales also moves quietly through the cells of your own body tonight. The same molecular patience that carried traits through forgotten generations is still at work beneath your skin, faithful and unseen. Perhaps that is why genetics feels so haunting in the quiet hours. It speaks to intimacy and scale at once. It tells us that life is historical, that each living being is both an arrival and a continuation, both something new and something carried forward. We inherit patterns, but we also live them in singular ways. The code persists, yet no life is merely a copy. So, as the room grows softer and the day recedes, let the details dissolve if they wish. The names can drift, the mechanisms can blur. What matters now is only the feeling of the thing, that beneath breath and blood and sleep there is order, and beneath that order there is memory, and within that memory there is room for change. A hidden grammar written into matter, a long continuity moving quietly through time, quietly, patiently, through every living thing. And for tonight, that is enough. Good night.